Hello everyone. Welcome to my YouTube channel. Today our topic is Fluid Mosaic Model of Membrane Structure. The cell envelope is defined as the plasma membrane and all the surrounding layers external to it. The cell envelopes of many bacteria consist of the plasma membrane, cell wall, and at least one additional layer, for example, capsule or slime layer. Of all these layers, the plasma membrane is the most important here. We will discuss the bacterial plasma membrane. Plasma membrane is the most important because it encompasses the cytoplasm and defines the cell. The plasma membrane is responsible for much of the cell's relationship with the outside world. Plasma membranes are an absolute requirement for all living organisms because they are involved in carrying out these cellular tasks. A primary role of all plasma membranes is that they are selectively permeable barriers. They allow particular ions and molecules to pass either into or out of the cell, while preventing the movement of others. Bacterial plasma membranes play additional critical roles. They are the location of several crucial metabolic processes: respiration, photosynthesis, and the synthesis of lipids and cell wall constituents. To understand these chemical differences and the many functions of the plasma membrane and other membranes, it is necessary to become familiar with membrane structure. The most widely accepted model for membrane structure is the Fluid Mosaic Model. Fluid Mosaic Model of Singer and Nicholson, which proposes that membranes are lipid bilayers within which proteins float. Here you can see that membrane is composed of two sheets of lipid molecules arranged end to end. The chemical nature of membrane lipids is critical to their ability to form bilayers. Most membrane-associated lipids are amphipathic. They are structurally asymmetric, with polar ends and nonpolar ends. The polar ends interact with water and are hydrophilic, while the nonpolar hydrophobic ends are insoluble in water and tend to associate with one another. The outer surfaces of the bilayer are hydrophilic, whereas hydrophobic ends are buried in the interior away from the surrounding water. Membrane proteins. Two types of membrane proteins have been identified based on their ability to be separated from the membrane. The one are peripheral membrane proteins and the other one are integral membrane proteins. Peripheral membrane proteins are loosely connected to the membrane and can be easily removed. They are soluble in aqueous solutions and make up about 20 to 30% of total membrane protein. Integral membrane proteins. These are not easily extracted from membranes and are insoluble in aqueous solutions. Integral membrane proteins carry out some of the most important functions of the membrane. Many are transport proteins used to move materials either into or out of the cell. The Fluid Mosaic Model of Bacterial Membrane Structure. This diagram shows the integral membrane proteins blue floating in a lipid bilayer. Peripheral membrane proteins purple are associated loosely with the inner membrane surface and integral membrane proteins. Small tan spheres represent the hydrophilic ends of membrane phospholipids and wiggly tails are the hydrophobic fatty acid chains. Phospholipids are drawn much larger than their actual size. Oligosaccharides, the chains of carbohydrates, protrude into the environment and may be attached to proteins or to membrane phospholipids. If you like this video, then hit the like and subscribe button and share it with your friends. Thank you.